This is part 2 of our urinary lecture. We're going to pick up where we left off on our previous unit, so labeled part 1. We're going to talk about tubular reabsorption and secretion. So first we're going to review exactly what's happening, and we're going to look at in depth what is being reabsorbed, what is being secreted, and exactly where and how these actions are partaking. So first and foremost, we have to realize that much of the filtrate is going to be reabsorbed, especially the water. If you go back to our last lecture, we talked about the amount of filtrate, that 150 to 180 liters a day, that amount of filtrate that is being produced by the renal corpuscles, specifically that glomerulus, and how we introduced a word called obligatory water reabsorption, how we have to pick up majority, 90% of that water is going to be reabsorbed right here at that beginning portion of that proximal convoluted tubule. So when we say that especially water right here, especially water is going to be reabsorbed. So much of that filtrate being reabsorbed, especially water. We will also discuss glucose and how glucose is going to be picked up back here. We mentioned and hinted to diabetes and how an excess of sugar in the blood, that our kidneys can't possibly reabsorb all that glucose, and it's a small enough molecule to make it through that pore system of the glomerulus. So we do see it when we do urinalysis, see it in urine being excreted in urine. The third thing we pick up or reabsorb is going to be amino acids, and the last thing are going to be some ions. And again, we'll elaborate on this a little further on in this lecture. As far as secretion is concerned, secretion is going to help manage the pH of urine, keeping it pretty acidic, and it's going to help rid the body of toxics and foreign substances, things like drug metabolites, etc. Plasma filtrate and urine compositions. Table 26.3 does an excellent job to explain and outline the substances filtered, what's being reabsorbed, and what's being secreted per day. So this gives us a pretty good idea of how much and how many things are going to be picked up back, reabsorbed back, and brought back to the blood as opposed to being secreted in our urine. So we start with our first one, which is water. We see that we're filtering about 180 liters, or we're getting about 180 liters of filtrate. And then here's that reabsorbed. Look at that. 178 to almost 178.5 liters is being reabsorbed back into the blood. So again, we're talking about blood volume. We have to maintain consistency of our blood volume. So it's important that we pick up all this water, all this plasma, all this filtrate again. We're going to secrete about 1.5 to 2 liters, okay, per day. We look at protein. Protein gives about 2 grams of protein is going to be filtered a day. We reabsorb 1.9. We secrete 0.1. So we will look at albumin levels later on when we do urinalysis. Sodium ions, about 579 grams is going to be filtered, and about 575 is going to be picked back up or reabsorbed back into the blood, and about 4 grams is going to be secreted in our urine. Chloride, 640 to reabsorb 633, and then secreted about 6.3 grams. Bicarbonate ion, we take almost 99% of the bicarbonate back. So bicarbonate's about 27.5 grams. We reabsorb 27.4.97, 274.97, and we secrete 0.03 grams. So it's a massive amount of bicarbonate. Again, our buffer to our blood system is going to be reabsorbed almost in its entirety. Glucose, we're taking all of it. And again, this is assuming normal conditions. 162 grams of glucose is going to be filtered, and 162 is going to be reabsorbed, and 0 is going to be secreted out into the urine. Now again, assuming that we are not diabetic and we don't have excess sugar in our blood, if you have a person who's diabetic and have high amounts of sugar, you will find this number here, the amount of sugar in the urine, will be elevated. As far as urea goes, 54 grams is going to be filtered, 24 grams reabsorbed, and 30 grams are going to be secreted back into your urine. So again, this is that waste product from digestion that was moved to the liver as ammonia, that was converted to urea, and then carried through and back and secreted out into our urine. The next substance on our list is potassium. So we get about 29.6 grams of potassium filtered, and we are picking up 29.6 back reabsorbed. The secreted value can vary. So it shows that we get secreted back into our urine about 2 grams, but again, pretty much all the potassium is reabsorbed. And again, this can vary depending on the end portion, the distal convoluted tubule and those principal cells in the collecting duct, where we're going to see the real fine tuning. So again, the amount of potassium can be variable, and it depends on the hydration and hormones in the body, specifically aldosterone and antidiuretic hormone. Next is uric acid. 8.5 grams of uric acid, 7.7 is going to be returned to the blood, and 0.8 is going to be secreted. Creatinine, 1.6 grams. So we go back to muscle metabolism, the waste product of muscle metabolism. We see that 0, and this is pretty interesting, 0%, this is a waste product, 0% is going to be returned to the blood, and almost all of it, 1.6 grams, is going to be excreted in our urine. And we can do creatinine clearance tests when we do urinalysis to see exactly how much of that metabolic waste product of muscle metabolism is really getting released and removed from the body. Reabsorption routes. So when we talk about and elaborate on reabsorption, we looked at the previous table and explained what's being reabsorbed, what's being secreted. We have to delve a little bit more into exactly how do these substances get reabsorbed through these tubules. Well, there's two basic processes. The first being paracellular reabsorption. This is going to be the passive leakage of fluid between the cells. The second is going to be transcellular reabsorption. So trans meaning across, cellular meaning the cell. So this is going to allow for stuff to move directly through the cells of those tubules. This picture does a pretty good job of explaining and showing and demonstrating our two different reabsorption routes. We start with paracellular reabsorption, and we see that sodium is moving. So we really have to understand how sodium is moving. So basically what we can think is how and where paracellular between cells can sodium move. And we go back to concentration gradients. This is a passive process where we're going to move sodium down a concentration gradient. This is different from transcellular. Transcellular transport involves energy expenditure, right? So this is a little bit different. We need ATP. We need that conversion from ATP to ADP to power these gates to open and move sodium through. So the nice thing about this paracellular transport is that the absorption rate is pretty much going to be matched to the load. And the reason why is because there's no transporters that can be saturated, unlike in transcellular reabsorption. We'll get into those transporters, the antiporters, the symporters, and discuss how they work. So again, we deal with diffusion, moving across right here, moving across a concentration gradient. We deal with active transport, so again, energy expenditure to get the sodium typically moving against a concentration gradient in some cases. And again, our sodium potassium pumps, so these are a 3:2 ratio. Again, energy expenditure to get it into this paratubular capillary. We can look at transport mechanisms, specifically the ones that require energy. So we have two different types of active transport, first being primary, the second one being, well, secondary. For primary transport, we're definitely using ATP. This is going to be our sodium potassium pumps. Now, the nice thing is that at rest, this is going to account for about 6% of our total body ATP use. So we're not using a majority or a massive amount of our ATP production, but a small percentage to help our kidneys work to produce our urine, especially while we're resting or sleeping. Our secondary active transport, this is going to be driven by ions' electrochemical gradients. So we need specific channels like symporters and antiporters. Now the difference between these two channels are symporters are going to move substances, sim meaning in the same, so in the same directions, anti meaning against, so our antiporters are going to move in the opposite direction. So again, symporters are going to move in the same direction, where antiporters are going to move substances in an opposite direction, typically against a concentration gradient. That brings us to water reabsorption. Now, we talked about obligatory water reabsorption occurring in the proximal convoluted tubule and portions of the descending limb. And we introduced this word as being obligated or obligatory, right? So what this means is it has to. And this is that massive amount of water has to be picked back up or reabsorbed back into the blood. 90% follows. So as soon as it's filtered, that 150 to 180 liters of filtrate, as it's moving through the glomerulus and into or through that capsular space into that first portion of the distal convoluted tubule, right away we're going to start reabsorbing water. So water has to follow those solutes that are being reabsorbed. And again, here, has to, obligated, 90% of that water is going to be picked back up. Now we say, well, where's the other 10% picked up? Well, this is going to be called facultative water reabsorption. Again, we think about the terminology. We think about what this word facultative means, right? So facilitate or faculty. We think what faculty's job is to help disseminate information to you and to get you to understand what is going on in these topics, kind of like what we're doing now. So facultative water reabsorption is an assisted type of water reabsorption. 10% of water is going to be picked up via this manner. We're going to allow water to be reabsorbed via this process. So then we have to really understand it's regulated by hormones. The hormone that's regulating this reabsorption is antidiuretic hormone. So we say, well, how does this do that? We go back to our endocrine lecture and we start thinking. We say, we know that antidiuretic hormone is secreted when the body is in states of dehydration. So exactly what's happening and who is this happening to? So we're talking about the latter portion of our nephron. We're talking about and we could write this down. So for obligatory, we're talking about our proximal convoluted tubule and our descending limb. For facultative, we're talking about our distal convoluted tubule and our collecting ducts. Okay? So when we're talking about what is in these collecting ducts and what is in this distal convoluted tubules, we're going to see these channels, the channels that are going to help for the reabsorption of water when called for. And then we say, well, who's calling for the reabsorption of water? Who's regulating this 10% of water that may or may not be reabsorbed? And the answer is right here. It's antidiuretic hormone. So here's a scenario. We're walking through the desert. We're in a state of dehydration. The hypothalamus is going to detect that. It's going to talk to the posterior pituitary that's storing all this amount or these high amounts of antidiuretic hormone. It's going to move through the blood and it's going to go talk to the kidneys and specifically talk to these tubules of these portions of the tubules. And what it's going to say is, listen, we need to reabsorb more water. We need to maintain blood volume. We need to maintain the hydration in the interstitium and in the tissues and the space and the fluid between our cells. So it's essentially going to open up the channels, and we'll name these channels in a little bit, these aquaporin channels. It's going to open up these channels, allowing for water to be reabsorbed. If the body is well hydrated, we do not see this 10% of facultative water reabsorption occurring. Technically, it is shut down until we detect, or parts of our body detect, low blood volume, dehydration, high sodium chloride ions, etc., etc., etc. We're going to slowly focus on each and every portion of this nephron to see exactly what is moving in and out and how it is moving in and out. So we start first with our sodium glucose symporters or cotransporters. So this is located pretty much exclusively to the proximal convoluted tubule. And again, its job is going to be responsible for the reabsorption, of all that filtered glucose, okay, that just occurred at the nephron. The second membrane protein that we're going to look at is the sodium hydrogen antiporter. The job of this is going to be to transport sodium into the cell and hydrogen out of the cell. This is where that antiport word is coming from. Now, we specifically find this in the proximal convoluted tubule. We're also going to find some in the collecting ducts of the nephron. Right? But in the proximal convoluted tubule, its main goal, its main job, its primary responsibility is going to be to maintain homeostasis of pH, that's where these hydrogen ions come from, as well as sodium concentrations. Now, where this can get interesting is from a clinical aspect. If we get defects in this, let's change the color just so we can denote a clinical term. So if we deal with the clinical aspects of these sodium hydrogen antiporters, and we say, well, what happens when they become defective? Well, this can have drastic effects on your heart or kidney. What happens is we lose the ability to maintain and manage pH, lose the ability to maintain and manage sodium, and as a result, we could end up with heart or kidney failure. That brings us to our last membrane protein, and that's aquaporin 1. This protein is going to be a water-specific channel, and what it's going to do is it's going to provide the plasma membranes of the kidney's proximal convoluted tubules, because that's where we're working. We're again in the PCT. So it's going to allow the cells in the proximal convoluted tubule to become highly permeable to water. And basically what this means is that water is going to be able to move in a single direction. Right? And that direction is going to follow an osmotic gradient, allowing it to be reabsorbed back into the blood. Our next slide shows the actual transport mechanisms. Everything we just talked about, but just putting it to picture, looking at both the symporters and our antiporters. So again, pay note that we are focusing in right here at our proximal convoluted tubule. That's where we're staying. And we can see that we have sodium moving in, we have glucose moving in, so we're using our sodium glucose symporters to help those move through. And then on the opposite end, how is this getting picked up back into our peritubular capillary, getting reabsorbed? And we see, we're going to switch over, we'll keep this red here, and we'll see that we're using glucose facilitated diffusion. And this transporter is going to help transport glucose from inside the cell, move it to the interstitial fluid so that it can be picked up by that peritubular capillary that was, and if you remember, sweeping around those tubules, okay? So then we could go through our sodium-potassium pumps and we say, well, how do we get this sodium that was just brought in? So 2 sodiums for every 1 glucose is brought in with our symporter. How do we get it into the interstitium to get picked up again by this capillary? Well, we need energy, and that's where our ATP comes in. So we're going to use our active transport, our sodium-potassium pump, to utilize that, release of phosphate, that energy to open it up, drop sodium into the interstitium, allowing it to be picked up by that peritubular capillary. The next half of this is going to be our antiporters. You know, again, we're talking about moving sodium. So if we look here, we'll change colors. We'll move sodium into the cell while moving hydrogen out of the cell. So again, let's erase that so that we can see it. My writing muddled it a little bit, but we're dealing with that sodium hydrogen exchange. So we have metabolic reactions going on in here. This metabolic reaction should look somewhat familiar to you. So let's go in here. We're talking about cellular respiration. We're going to keep our green, and we're going to see that carbon dioxide is going to be moved into the cell. Now we know that our carbon dioxide is going to be converted. So we add water. We use our friendly enzyme that we recognize, and that's right here, carbonic anhydrase. So we get that catalytic reaction of carbon dioxide and water yielding this weak acid. Again, carbonic acid. Now we get that dissociation to bicarbonate. There's our buffer. And we're left with hydrogen ions. So that's where these hydrogen ions are coming from. And again, remember, we're moving this out of the cell. We're help maintain and manage pH, while at the same time bringing sodium in to help maintain sodium concentrations. Now that brings us to our sodium-potassium pumps. So we're going to zoom on out, and we're going to talk about exactly how we deal with sodium and what we do with that excess bicarbonate. So we're going to use facilitated diffusion to get that bicarbonate out of the cell into the interstitium and then back into that peritubular capillaries to be reabsorbed. Remember, we said that we are picking up nearly a little more than 99% of that bicarbonate, reabsorbing it back into the blood. And the sodium is going to be no different. We use a sodium-potassium pump right here. So sodium-potassium pump, so again, requiring energy. So we see ATP into ADP to get that sodium into the interstitium and then sucking it back into our peritubular capillary. So the next question is, well, where did this CO2 come from? Well, it came from two places. It came from one inside the actual tubule, so what was dissolved in the filtrate. Again, remember, we're at the proximal convoluted tubule, so we're right at the beginning of this highway working its way to concentrate and make urine. And then we're getting some CO2 from the peritubular capillary that's coming in. Two places, all contributing to the formation of this bicarbonate ion. Now the picture at the bottom is essentially the bicarbonate being reabsorbed, and we covered that up here, right? We said that we're using carbonic anhydrase, and we are converting this carbon dioxide and water to carbonic acid, and then releasing a hydrogen that's working with our antiporters, dealing with bicarbonate, and using facilitated diffusion to get it into the blood capillary. But what we didn't talk about was this conversion here. So inside that lumen, right, the fluid in the proximal convoluted tubule and the actual lumen, we have some bicarbonate. And we're using that hydrogen ion here to catalyze with carbonic anhydrase to catalyze the conversion to our weak acid, right? So we talk about carbonic acid, again, breaking it into a water and a carbon dioxide. And that carbon dioxide is going to diffuse its way into the cell, contributing to this formula that we talked about a few seconds earlier. As we continue to move down this tubule, and we're entering into the late portion of the proximal convoluted tubule, we can start looking at some more things that are constituents that are moving from the fluid, from the stuff, the solution, the filtrate in the tubule, in that lumen. And we see that we're dealing with a few ions: chloride, potassium, calcium, magnesium, and urea. It is all going to be diffused, so we use diffusion. And look at the routes. We're going through and in between the cells. We're not relying on any complex active transports, no symporters, no antiporters. We're simply diffusing into the interstitium and then reabsorbing it here in the peritubular capillary. The last substance we have to look at that's being reabsorbed is going to be water. And we see that water is going to move via simple osmosis, right? This is still part of our obligatory water reabsorption, and then reabsorbed back into the peritubular capillary. The next portion of this highway, or this nephron, or the loop that we have to talk about, is reabsorption in the loop of Henle. Exactly what's going on in that downturn as it makes its way up. So we see, number one, that we have relatively very little water reabsorption. So we're relatively impermeable to water, especially in the ascending limb. And if we recall, we talked about this before, where on the upturn, the ascending limb got significantly thicker. And we said it got thicker because we were going to naturally block water, not allow it to be reabsorbed. So let's erase this because we will look at a picture of this to make more sense of it. So again, we're relatively impermeable to water here, especially in the ascending limb. We have very little obligatory water reabsorption here. Okay, and this is going to follow suit with this impermeability, relatively impermeable to water. And then the last thing we're going to see is our sodium, potassium, and chloride symporters. So our next slide is going to show us a picture to explain exactly how this works. Now this picture does a good job to show us an in-depth look on the loop of Henle and reabsorption routes. So what we're going to focus on is right here, this sodium chloride potassium symporter. So again, the point of this membrane protein, transport protein, is going to be to move sodium, two chlorides, and a potassium across the cell membrane. Now they are moving in the same direction, which is why it's considered a symporter. But a key note, I want you to look. We have one positive, two positive, and two negatives. An important part of this symporter is to maintain electroneutrality, and it does this by moving two positively charged solutes, sodium and potassium. So we have that sodium and the potassium, so a plus and a plus make the two pluses, and it's going to move it alongside two parts of a negatively charged ion, this being chloride. So again, they cancel each other out, so we do not affect the electronegativity of the solution in the fluid of the tubule as it moves into the cell. Once inside the cell, we have to figure out how to reabsorb this back into the vasa recta. So notice we're now no longer saying peritubular capillaries, we're talking vasa recta. So in your mind, you should be thinking about these long juxtamedullary nephrons. And we are focusing in that ascending limb of the loop. So we're coming down and up with this solvent and the solutes. So exactly what's happening. So we're using sodium potassium pumps to take all that sodium, move it into the interstitium, and then reabsorbing it back into the vasa recta. We're using leak channels. So again, leak channels that are going to distribute those chlorides and then put it into the interstitium so that it can be reabsorbed again by the vasa recta. Now I want you to note the potassium. The potassium is going to simply leak in and out of the cell to maintain that electroneutrality. But now notice, no potassium is moving out of the cell to be reabsorbed back into the vasa recta. So now we have an imbalance, an electrical imbalance, because we have two negatively charged ions and one positively charged ion. What this does is it's going to set up that interstitial fluid to be more negative than the fluid inside the lumen of the tubule. That brings us to our next step down here, our cations. We are going to move some cations with simple diffusion, and we're going to take sodium, potassium, calcium, and magnesium, and we're going to simply move them. So all that excess potassium, calcium, magnesium, and sodium is going to move right into that vasa recta. Now one last thing, look at the thickness of this apical surface of this cell. Now this is going to be lined with a glycoprotein. That glycoprotein is going to be very hydrophobic, so it's going to block any water from moving through the cell and being reabsorbed back into the vasa recta. We do not want water being reabsorbed here. We want to keep the urine relatively dilute, and especially in this structure because we have to deal with the countercurrent exchange mechanism. So this is going to set up or help to set up the countercurrent exchange mechanism that we're going to talk about in a little bit. The next portion of the tube that we're going to look at is going to be reabsorption in the early distal convoluted tubule. We're going to see that we're going to be moving ions from the tubular fluid into the cells of the DCT. We do that with these sodium chloride symporters or co-transporters. They're going to move both sodium and chloride into the cells. We're also going to see hormonal action here. We're going to see parathyroid hormones going to stimulate the reabsorption of calcium. At the same time, it's also going to inhibit phosphate reabsorption in the proximal convoluted tubule. What this does is basically enhance its excretion. In the late distal convoluted tubule and the collecting duct, we're going to see three primary actions. The first are going to be principal cells. These are the main sodium reabsorbing cells and the site of action of aldosterone. So when we talk about aldosterone being secreted from the adrenal gland and what it does regarding sodium and potassium management, we talk about these specific cells, these principal cells, as the site of action, and the location is going to be the distal convoluted tubule and the collecting duct. We're also going to see that the principal cells are going to secrete potassium through an apical membrane potassium channel. The second structure we're going to find is aquaporin 2. This protein forms a channel that's going to carry water across the cell membrane. It is found in the late DCT as well as the collecting duct. These are going to be stimulated by antidiuretic hormone secreted from the hypothalamus and the posterior pituitary. The third and last cell that we're going to find in the late portion of the distal convoluted tubule and the collecting duct are called intercalated cells. Their job is going to be to mediate acid secretion and bicarbonate reabsorption. So we're monitoring secretion of hydrogen ions. This is going to be our acid secretion. And then the primary focus is going to be here in the reabsorption of bicarbonate. We're also going to reabsorb some potassium here as well. Now we can put this all together by looking at a negative feedback loop which regulates water reabsorption with the aid of antidiuretic hormone. So we start right here at the stimulus. So the stimulus is going to basically disrupt homeostasis by increasing osmolarity of plasma within that interstitial fluid. So that's that controlled condition that we are changing. The osmoreceptors in the hypothalamus are going to detect this increase, at which point it's going to talk to the control center, that hypothalamus and the posterior pituitary, to secrete ADH. ADH is going to move through the blood and it's going to attach an effect to those principal cells. Those principal cells, once ADH has attached to them, become more permeable to water, which is going to increase the second part. Remember, we talked about obligatory water reabsorption and facultative water reabsorption. Right here, we're going to utilize that 10% of water reabsorption via facultative water absorption, and again, due in fact to the binding of ADH to those principal cells. As a response, we get a decrease in plasma osmolarity as we increase water. We return back to homeostasis, the system shuts down, and we're back to normal. Table 26.1 discusses the distal collecting tubule and collecting ducts and looking at the cell types, description, and its region and histology. So at the proximal convoluted tubule, we see an apical surface, we see mitochondria, we see microvilli. So when we outline it, we say it's simple cuboidal epithelium with a prominent brush border of microvilli. We think back to our modifications. What's microvilli for? Absorption. So this is going to be one of our modifications to help us absorb in the proximal convoluted tubule. The nephron loop, the descending limb and the ascending limb, look at the change. It's conformational change. We go from cuboidal to simple squamous. We also lose those microvilli, so less reabsorption is occurring here. In the nephron loop, then the thick portion, remember we get thicker, we get simple cuboidal epithelium to low columnar epithelium. So we're getting thicker and thicker, again getting to the point where we're going to line this with the glycoprotein to block water reabsorption altogether. Most of the distal convoluted tubule is going to be simple cuboidal epithelium. The late part of the DCT and all of the collecting duct, we're going to see simple cuboidal epithelium. We zoom on in. We see our principal cells, that's going to bind ADH. We see our intercalated cells, that's absorbing. So look at those microvilli again. We're going to be reabsorbing that bicarbonate and secreting, doing all that fun stuff with bicarbonate and potassium and hydrogen ions. So in our next few slides, this is basically going to be a quick review and summary and putting together all the important facts of what's going on a basic function, on a basic functional level of the anatomy of the nephron. So we kind of put it all together in the next few slides. So on the PCT, what's occurring? We're getting reabsorption of 70% of that filtrate that was just made through the glomerulus. we are getting 100% reabsorption of glucose, and this is what we said in our previous tables, and our amino acids. We are doing this under obligatory water reabsorption. There's our process. As far as the arrangement, we get simple cuboidal epithelium with microvilli. That's what's going to allow us to reabsorb. We utilize carrier transport proteins and mitochondria to help us move other things, ions, in and out of the cell. In the loop of Henle, we're going to see the descending limb is going to be thin, water is going to leave, and solutes are going to concentrate. In the ascending limb, the thicker limb, remember we have that glycoprotein blocking water, water stays, solutes are going to be diluted. And we will look at a chart to see the difference in concentrations as we concentrate and dilute that urine moving down through that loop and back up. We look at the distal convoluted tubule. The DCT is going to actively secrete ions, toxins, and drugs. We're going to reabsorb sodium ions from our tubular fluid. And water and sodium reabsorption in this area is going to be mediated by hormone secretion. The two major hormones are ADH and aldosterone. And by now you should be thinking about the specific cells that these guys are attaching to essentially affect that water reabsorption. Hormonal regulation of tubular reabsorption and secretion. Table 26.2 does a really good job in putting all these hormones together, the major stimuli that trigger the release, the mechanism and site of the action, and the overall effect. So we could go through this. We have talked about this before, but again, this kind of puts it all into motion. We talk about angiotensin 2, where the stimuli that's being detected is low blood volume or low blood pressure. It's going to stimulate the renin-induced production of angiotensin 2, so our RAA. The mechanism and site of action is stimulate activity of sodium hydrogen antiporters in the PCT and the proximal convoluted tubule. The overall effect, increased reabsorption of sodium, so increasing sodium reabsorption, and water reabsorption, which is going to overall increase blood volume and blood pressure. Aldosterone. Aldosterone is going to be seen major stimuli when an increase in angiotensin 2 levels and increased level of potassium plasma promote the release of aldosterone by the adrenal cortex. Overall mechanism, we're going to enhance activity of those sodium-potassium pumps in the basolateral membrane and sodium channels in the apical membrane of our principal cells. Here's our principal cells again in the collecting duct. Overall effect, increased secretion of potassium and reabsorption of sodium, increases reabsorption of water, which increases blood volume and blood pressure. See the goal here? Increasing blood volume, blood pressure again and again. ADH, antidiuretic hormone, stimuli, increased osmolarity of extracellular fluid or decreased blood volume, again due to dehydration or similar, is going to promote release of ADH from the posterior pituitary. Overall goal, mechanism of site of action, stimulate insertion of water channel proteins. Here's our aquaporin 2 channels into the apical membranes of principal cells. This is going to increase, look at our terminology, facultative reabsorption of water, which is going to decrease osmolarity of body fluids, essentially rehydrating. AMP, atrial natriuretic peptide, major stimuli, stretching of the atria of the heart is going to stimulate AMP secretion. Its mechanism, it's going to suppress reabsorption of sodium and water in the PCT and the collecting duct, and this is going to inhibit secretion of aldosterone and ADH. Overall effects, increase excretion of sodium in the urine. This is natriuresis, which is going to increase urine output, diuresis, and thus overall decreasing blood volume and blood pressure. Remember, we talked about AMP being the opposite. It was the only one that was decreasing blood volume and blood pressure. All the other ones were increasing. Okay? And then the last one is parathyroid hormone, which is going to detect decreased levels of blood calcium, which is going to stimulate PTH from the parathyroid gland. This is going to stimulate openings of calcium channels in the apical membrane of our distal tubule, our distal convoluted tubule, and as an effect is going to increase reabsorption of calcium back into the blood. So far, we have talked about the nephron and exactly what is moving in and out of the cell, in and out of the blood, in and out of that tubular fluid. But what we haven't talked about yet is actual urine production and how it's occurring, where concentrations are varying. So that's what we're going to cover in our next few slides. So for urine production, we have to know a few things. Fluid intake is going to be highly variable, depending on what you're taking in your body through digestion, through drinking, etc. So this is going to directly affect urine production. Homeostasis requires maintenance of fluid volumes, but they have to be within specific limits. And urine concentration will vary, especially when exposed to antidiuretic hormone. So when we talk about fluid intake, we'll see two basic premises. Number one, high intake of fluid is going to result in dilute urine. And we see this during the day as you're drinking water and juice and all this stuff. We see that our urine is going to be a light pale yellow. So that high intake of fluid gives us that dilute urine. If you think about when you go to bed, you're sleeping, you're not drinking much, so you have a low intake of fluid. That urine is going to be concentrated, so we have concentrated urine of low volume. So again, wake up as soon as you go to the bathroom in the morning, look at your urine. It is dark yellow. And again, this is going to be that highly concentrated urine as your body's conserving fluids while you're sleeping. So for formation of dilute urine, we look at glomerular filtrate and blood. They're going to have about the same osmolarity as at about 300 milliosmol per liter. However, tubular osmolarity changes due to a concentration gradient in the medulla. So that's the key to allowing this system to work. The medulla has to be maintained at a super salty concentration. This helps set up our concentration gradient, as well as a countercurrent exchange mechanism that we will talk about in a little bit. For formation of dilute urine, we see a couple scenarios occurring. So when dilute urine is formed, osmolarity in the tubule is going to increase. So we're going to increase in the descending limb, and we're going to decrease in the ascending limb and decrease some more in the collecting duct. And we're going to show a nice little chart to look at the concentrations and the osmolarity as we're moving through the tubules in a little bit. Formation of dilute urine in the two later portions of our nephron in the thick ascending limb is going to be regulated by symporter activity, which is going to be reabsorbing sodium, potassium, and chloride, dependent on a low water permeability aspect, where solutes are going to leave and water is going to be staying inside that tubule again. So we see another mechanism to aid in that countercurrent exchange. And again, we're throwing this terminology in, but we haven't discussed it yet, so hang on, we will. In the collecting duct, we have very low water permeability, especially in the absence of antidiuretic hormone. Remember, antidiuretic hormone is going to open this up to increase water permeability. Now we have a picture. We're putting this all to picture now so we can see some osmolarities and what's occurring. So we're going to switch over to our black pen. We see that blood is coming in. We're going to be filtering, and our concentrations about 300 milliosmol. As we move down, remember, water is being reabsorbed here via obligatory water loss. And what we see is as we continue this trek down, concentration of urine is increasing. And this is to be expected. We expect it to increase. Water is being reabsorbed. Now, as we hit the bottom of that loop, the majority of water, 80%, 90% of that water was pulled back up, so it could be 80 to 90%, was pulled back up, was reabsorbed back into the peritubular capillaries, at which point, remember, look at the thickness. Look how thick this ascending limb is. Remember, we are blocking water reabsorption, but allowing solutes to leave. So, now what happens? As we continue up this loop, we're blocking water from leaving, but we're kicking some constituents out. So we start dropping back down in osmolarity. Right? So we're making this dilute urine. Once we get into the collecting duct, this is really where we're going to be fine-tuning our dilute urine, fine-tuning the constituents of it. And we see a further drop to about 65 milliosmol for a concentration of our urine. So now we could switch over from dilute urine to our formation of concentrated urine. Now we're going to go back a little bit and refer back to our talk where we discussed the two different types of nephrons, cortical nephrons and juxtamedullary nephrons. And if you recall, our juxtamedullary nephrons were those longer loops that extended the majority of the nephron into the medulla, whose job was primarily to deal with concentrating urine. And they're going to do this by maintaining and adjusting osmotic gradients. Now, this osmotic gradient is going to be created by something called the countercurrent multiplier. So I promised you that we were going to discuss it, and this is where we're going to do that. So the goal of the countercurrent multiplier is to maintain a super salty concentration within the medulla. So the medulla osmolarity must be increased. We're going to do this via a process of 7 mechanisms. Essentially, solutes are pumped out of the ascending limb, but water is going to stay in the tubule. We're going to look at this mechanism through 7 steps. Step 1. We're going to assume that the loop of Henle is going to be filled with a concentration of about 300 milliosmol per liter. This is going to be the same as what's leaving the proximal tubules, the proximal convoluted tubules. Step 2. The active ion pump of the thick ascending limb on the loop of Henle reduces the concentration inside the tubule and raises the interstitial concentration. Step 3. The tubular fluid in the descending limb and the interstitial fluid quickly reaches osmotic equilibrium, and this happens because of osmosis. Water is moving out of that descending limb via obligatory water loss. Step 4. The additional flow of the fluid into the loop of Henle from the PCT, which causes the hyperosmotic fluid previously formed in the descending limb to flow into the ascending limb. Step 5. Additional ions pumped into the interstitium with water remaining in the tubular fluid. We're going to do this until about 200 milliosmol per liter osmotic gradient is established. Step 6. Now again, the fluid in the descending limb reaches equilibrium with the hyperosmotic medullary interstitial fluid, and as the hyperosmotic tubular fluid from the descending limb flows into the ascending limb, still more solute is continuously pumped out of the tubules and deposited into the medullary interstitium. Step 7. These steps are repeated over and over again with the net effect of adding more and more solutes to the medulla in excess of water. So we're removing water or eliminating water and adding more and more solutes. With enough time, this process gradually traps solutes in the medulla and multiplies the concentration gradient, established by the active pumping of ions out of the thick ascending limb. Eventually, this raises the interstitial fluid osmolarity to 1200 to 1400 milliosmol per liter, essentially again making this super salty medullary interstitium. This is the countercurrent exchange mechanism. Continuing with formation of concentrated urine, we go back to antidiuretic hormone. So again, to review, in the presence of antidiuretic hormone, our collecting ducts become very permeable to water. As a result, tubular fluid becomes very concentrated as we move water back into the interstitium. Movement of water also carries urea into the medulla, contributing to its osmolarity. When we look at urine production, the main focus and goal is to maintain homeostasis. It does this by the secretion of waste into the tubular fluid, which is going to help to regulate 1, blood volume, and 2, normal blood composition. One of our first waste products that we see is urea. This is going to make up about 60 to 90% of all nitrogenous materials. And we say, well, where does this come from? When we talk about digestion, we'll talk about the deamination of protein. So we're breaking down our amino acids. It's going to move all the breakdown, the waste product of this breakdown of amino acids in the form of ammonia to the liver. The liver is going to combine this ammonia with carbon dioxide, and it's going to form something a little more friendlier for the body, which is urea. The second waste product we're going to see is going to be creatinine. This is derived from creatine phosphate in muscle tissue. So we're talking about muscle metabolism, the waste product of muscle metabolism, specifically creatine phosphate. On a typical normal person, we should be producing about 18 grams per day. And remember, this is a metabolic byproduct, a waste product. So 100% of it has to be eliminated. The last byproduct is uric acid. This is derived from the breakdown of nucleic acids from food or cellular destruction. Again, we're tying in digestion here. These tend to crystallize due to insolubility. This is a problem because this is a common component of kidney stones. So, If you have a high concentration of uric acid, they will crystallize and form these mace-like structures. So if you think about a mace ball, go back to ancient warfare, the balls with spikes on it. These crystallized portions of uric acid look like that, which is why when they get too big, they start tearing and ripping through portions of the kidneys, through the urethra, causing bleeding. And these kidney stones do have to be removed one of two ways. Either you'll pass them normally, or a doctor will go in with a either ultrasound or laser and sonically break up those crystals so that you can pass them in much smaller pieces. From talking to people who have had kidney stones, or if you know anybody who have had kidney stones, you know that this is a very painful process of passing these stones. So in review, so a lot of these slides help us put everything together and put a review format into what we're talking about with the formation of urine. We deal with one, filtration. Water and solutes move across the glomerular capillaries into that capsular space. The driving force behind filtration is the hydrostatic pressure of blood. So this is the heart beating. For reabsorption, we're talking about the removal of water and solutes from that filtrate back into the blood. Our body, our kidneys are master recyclers. We're recycling. All these key substances are reclaimed. Lastly, we have secretion. So we're going to be transporting solutes from the peritubular blood into the tubular fluid. Again, this tends to be drug wastes, drug metabolites, waste products, urea, etc. Renal function in review. So most regions of the nephron perform a combination of functions, as we said. General functions can be identified. I highlighted them in red to really bring out some of those main features and players. Filtration in the renal corpuscle occurs across the visceral border of the glomerulus. Nutrient reabsorption is going to occur along the proximal convoluted tubule mostly. Active secretion is going to occur at the proximal convoluted tubule and the distal convoluted tubule. The loops of Henle are going to regulate final volume and solute concentration of the tubular fluid. And this is going to be a great example of a positive feedback loop. That countercurrent exchange mechanism that we just talked about, we're going to see that the loops and duct cells require nutrients and oxygen from blood supply. Capillaries that feed them is the vasa recta. Remember, in the juxtamedullary nephrons, we have vasa recta instead of our peritubular capillaries. They form loops like those of the nephron loop in the medulla. Incoming and outgoing blood will have similar osmolarity. This is going to maintain medulla concentration gradient, so that hyperosmotic gradient that we just got finished talking about for the counter exchange multiplier. So we have a little 3D physiology, if you didn't like my drawing, that you can watch to explain the countercurrent exchange multiplier. This table does a great job to explain the overlap between blood, the blood vessel, the vasa recta, as well as the overlap of our nephron, of our tubule. So a couple key things to understand. As we increase our osmotic gradient from 300 to 1200, remember we're hitting that 12 to 1400 mark, we see that the concentration of the osmolarity of this solution inside the tubule is increasing to about 1200. Now remember that we said the blood vessel and the blood and the fluid osmolarity remains relatively constant. So look at the bottom of this peritubular capillary. We see that the osmolarity is, again, right about equal with that of the fluid within the tubule. The only difference is this countercurrent exchange mechanism. Again, what we should do is take this blood vessel, and I'm going to draw red to indicate that it should be lying over and around, flowing in and between these tubules. And what happens is, as this blood is flowing and as the fluid is flowing, we see that countercurrent exchange mechanism again. We see the symporters are going to be pushing out, we'll switch to green, pushing out sodium chloride in that thick ascending limb. We see the obligatory water loss coming out in the descending limb. And again, the reabsorption and the secretion through the blood capillary, through that capillary right here on the side, sodium moving in, water moving out, and on this opposite side, which If we drew a star here and correlated it to this region, we have water going in, sodium moving out. So again, what we're doing is this 3 times multiplier, where salt is moving out once, water is moving in 2, salt is moving out 3, and we get this increase in osmolarity in the interstitium. So this gets to be super salty, and this is good because it keeps the dynamics of this tubule working and going. This is one of my favorite tables. It summates pretty much everything that's going on in the entire nephron and puts it to color, so it makes it a little bit easier. So we can start at the renal corpuscle. We start right up at the top, right here, and we say that the glomerular filtration rate about 105 to 125 mils per minute of fluid that is isotonically balanced to blood. And then we talk about what's being filtered here. Water and all solutes present in blood is going to be filtered out, except for large proteins, including ions, glucose, amino acids, creatinine, and uric acid. So all these are moving out into the filtrate. Then we work at our proximal convoluted tubule right here, and then we can mark this number 2. We can see that what's being reabsorbed, so water 65%, the process, osmosis, sodium 65% via our sodium potassium pumps, utilizing our symporters and antiporters. We have potassium here 65% via diffusion. Glucose 100% is picked up via symporters and facilitated diffusion. Amino acids 100%, chloride 50%, bicarbonate 80 to 90% by facilitated diffusion, urea 50% by diffusion, and we'll see urea picked up again and secreted again through the nephron loop. Calcium, magnesium is going to be variable depending on what's in your body through diffusion. We talk about secretion in the proximal convoluted tubule. Hydrogen ions versus with our antiporters. Ammonia is going to be variable. We're going to see with cases of acidosis that it's going to increase, and we're using our antiporters. Urea is going to be variable again depending on your diet, and this is going to be diffusion. Creatinine, small amount. And then at the end of the PCT, we're going to see tubular fluid is still isotonic to blood, so at about 300 milliosmoles per liter. Then we go to our third portion, the loop of Henle, the nephron loop, right? And then we talk about reabsorption into the blood, so make sure we understand that we're reabsorbing into the blood. We're about 15% of water, this is osmosis. Sodium goes about 20 to 30% via symporters. Potassium, another 20 to 30%, again by symporters. Chloride is going to go 35% by symporters. Bicarbonate, another 10 to 20%. And this is going to be by facilitated diffusion. So if you look in total, we get about 80%, 85% of bicarbonate in the proximal convoluted. We get a little more right here in the nephron loop, another 10% in the nephron loop. And then we have calcium, magnesium, again variable through diffusion. What are we secreting now into the urine? So coming from the blood into the urine, we see urea, and we know that this is that metabolic byproduct of digestion. And again, this is variable, and we're going to recycle this from the collecting duct. And then at the end of the nephron loop, look at our tubular fluid. It is hypotonic, so it's going to be about 100 to 150 milliosmol per liter. Now we go right up here. We'll go to number 4, to our early distal convoluted tubule. We see reabsorption into the blood, another 10 to 15%. Sodium is going to be 5% by symporters. Chlorine, another 5% by symporters. Calcium, again, variable. And then that late distal convoluted tubule, so we're talking right around here and through this collecting duct here. Reabsorption of water, 5 to 10%. This is going to be through those insertion of those aquaporin 2 channels when stimulated by ADH, so this is facultative water reabsorption. Sodium, 1 to 4% by sodium potassium pumps, ATP-gated pumps. Then we have bicarbonate, again variable, depends on the acid or hydrogen ion secretion via the antiporters. Urea is going to be variable. We're recycling again to the nephron loop. Now the big one is going to be secretion. What are we secreting into the urine? Potassium. is going to be 1. Again, this is going to be variable. It's going to adjust for dietary intake. And then we're going to see some leakage channels here that's going to help push that potassium out. Hydrogen ions is going to be variable. It's going to move in and out with proton pumps, and this is going to help to maintain our acid-base homeostasis in the fluid and in the blood. And now the tubular fluid leaving the collecting duct is pretty dilute, especially when ADH level is low, and concentrated when ADH level is high. Well, that summates the entire nephron, what's being reabsorbed, what's being secreted. I hope you found this slide to be as helpful as I do. That brings us to evaluation of kidney function. This is something we're going to talk about in lab a little more, specifically urinalysis. So what we do as clinicians and doctors, and you've probably been to the doctor before for physicals or maybe regular blood work, that they will make you urinate inside a cup. What they're doing is they're evaluating your urine and checking for the presence of any abnormalities. So some things that we look for is albumin. Remember, albumin is a protein found in blood. It's relatively large and should not be making it through that glomerulus. If we do find that albumin has pushed its way through, the condition of having protein in your urine is called albuminuria. Okay, again, an abnormality. Glucose. We talked about glucose being pushed through the glomerulus, but then being reabsorbed 100% in the proximal convoluted tubule. But then we also alluded to the fact of people with diabetes, that there is way too much sugar in their blood that cannot possibly be reabsorbed in its entirety. So what we get is glucose in your urine. The condition is called glycosuria. Now, red blood cells. Again, red blood cells are relatively large. We would not expect to find red blood cells in our urine. If we do, it could indicate a whole host of disorders. Could be chronic high blood pressure, could be kidney failure. Something could have disrupted or ruptured one of the capillary bed inside of the glomerulus. So we're going to call this condition hematuria. Now, again, during urinalysis in lab, sometimes females will say that they find or test positive for blood in their urine and then get really nervous that this is a problem with their kidneys. Remember, know your cycle. If you are menstruating, so if you're undergoing menses, you will get some residual blood inside your urine. This is pretty normal. Typically in lab, we will be doing a true urinalysis test, meaning that you will urinate in a cup, and we will test it for pH, for specific gravity, for color, for smell, for whether we find albumin, glucose, red blood cells, white blood cells, which could indicate infection, urinary tract infections. The next thing we could look for are ketone bodies. Now, the condition, if we find a high level of ketones, is called ketonuria. And basically what happens here, we're detecting this elevated level of ketones. And what this can indicate is typically our body is burning fat stores. We're freeing fatty acids. And this can occur in times of fasting. So if you're fasting or if you're using specific types of diet, like where you're following a keto diet, where you're entering into ketoacidosis, you could have elevated ketones in your urine, etc. The last thing we talk about are microbes. Again, microbes found in the urine, this is something that we would look for microscopically, could indicate some type of urinary tract infection. So again, refer back to your lab book to go through all the different conditions, all the different constituents of urine, what elevations can mean from a clinical perspective. And again, we're going to review this in lab, so hang tight. Now that we have talked about and covered how we make urine, we have to talk about the transport. We kind of stopped at the kidney and didn't really go any further. So now we're going to look at where urine goes, where it's stored. So from the kidneys, from those series of larger and larger funnels, those minor calyx to the major calyx to the renal pelvis, we now are going to enter our ureters. Our ureters are going to transport urine from the renal pelvis by peristaltic waves. So these muscular contractions that's going to move the urine down using hydrostatic pressure and gravity into the bladder, our storage tank. There is no anatomical valve at the opening of the ureters into the bladder. So basically when the bladder fills, it compresses the opening and prevents backflow. The bladder is a relatively hollow, distendable, meaning it can expand and contract, muscular organ that has an average capacity of about 700 to 800 milliliters of urine. The ureters, the bladder, and the urethra in a female is shown in this picture. So we're going to go through and look at some of these structures and understand and try to work through how this system operates. So we're going to start with some basic general anatomy. Notice our ureters are working their way through the posterior aspect of the bladder, and they're going to enter right here at our urethral openings. Now, notice we have a triangle dashed here. This triangle is called the trigone. It is called trigone because it is triangular and it outlines this region from our internal urethral orifice to our two urethral openings. Now, these openings are circular here. As this bladder contracts, notice this big muscular wall, these will get more slit-like. And the reason why they get slit-like is because, remember, there is no valve. They have to close. They close with the expansion of this bladder to prevent urine from backflowing up the ureters. Now, as we leave the actual structure of the bladder, notice this big muscular wall. This is known as the detrusor muscle, and this muscle has a very important job. It's going to contract to help push urine into the urethra. Now, we have two muscles, sphincters, an internal urethral sphincter and an external urethral sphincter. So our first one is going to be right here. This little ring of muscle is our internal urethral sphincter. It is involuntary. It is smooth muscle. It's going to open and close based on a reflex of what's going on with the filling or the emptying of this bladder. The reflex that we're talking about is known as the micturition reflex. And we're going to go through this on the next slide. Now, below here is our external urethral sphincter. Now, the external urethral sphincter is skeletal muscle. This is under voluntary control, which is why when you feel that urge to urinate, that urge to pee, you can hold it until you get to a toilet. You'll release the contraction on that external urethral sphincter, and you will allow urine to flow out the urethra into your toilet. Now, the reflex that we were just talking about is the micturition reflex, and basically what this is involved in is discharging urine through voluntary and involuntary muscular contractions. So we're dealing with those two sphincters again. The internal sphincter is going to start spasming, it's going to start opening, and it's going to want to let the urine to leave, and this is detected and starts when that bladder is full, that 600 to 800 milliliters that we said before. Well, when it gets to that point, we're at the point where these muscles are going to open involuntarily and it's going to make you urinate. But now we're saved here because this reflex allows us to wait some time before we have to eliminate. Now what happens? We have stretch receptors that are going to trigger our spinal reflex. And we learn this in childhood, right? So what happens is at about 200 milliliters, that stretch reflex. So we have these receptors inside the detrusor muscle of the bladder that's detecting stretch. When it fills to about 200 milliliters, this receptor fires, sends a signal to the brain and says, I have to urinate or I have to pee. So basically what happens is our brain says this bladder is full, but not all the way full. It's up to 200. It's going to allow for almost a 4 times filling capacity before you're at the point of no return. What's next going to happen is that once we have this urge, this reflex to urinate, your body will eventually go through a cycle of contracting and releasing that internal urethral sphincter, at which point you're going to get to a toilet, you're going to control the external urethral sphincter and allow that urine to eliminate. Now the urethra is going to carry urine from the internal urethral orifice to the exterior part of the body. Now in males versus females, the difference is pretty much the length of the urethra. Males have a penis, so males have a longer urethra. Now, also, when we start talking about reproduction, we need to pay attention to our tubes. And when I say tubes, I mean our reproductive tubes versus our renal tubes or our urinary tubes. In males, we use one tube for reproduction and urination, and this can be problematic. So when we talk about reproduction, we'll talk about the structures that are in place to help protect the integrity or the viability of semen as it moves through this tube that has just carried waste and acidic natured urine. And we'll see that females have different tubes. Females have one tube for reproduction and one tube for urination, which can cause other problems, which is why females are more likely to get urinary tract infections, whereas males are less likely to get urinary tract infections. Well, that also includes the addition of another organ called the prostate, which also helps prevent males from getting urinary tract infections, which we will talk about when we do reproduction. Now we can use pictures to look at the male and female urethras, and this is pretty straightforward. The male urethra is obviously longer because we are traveling from the bladder through the length of the penis to the external urethral orifice. And we can break up this urethra into three regions. Region 1, where the urethra goes through the prostate, is called the prostatic urethra. Okay? And then we have this little urogenital membrane right here. Where the prostate moves through there, it's called the urogenital diaphragm. Where it moves through there, we're going to call it our membranous urethra. You can also use intermediate. We'll write membranous here. Membranous is a little bit easier to learn because it is moving through a membrane. And then the majority of the urethra right here is moving through the spongy portion, the corpus spongiosum, the spongy portion of the penis, so it is called the spongy urethra. You can also call it the penile urethra. Okay. Now for females, you notice one thing. There's no penis, obviously, right? So what happens is the distance from the bladder to the external urethral orifice is much shorter compared to that of the males. So if we look at the chart at the bottom, males versus females, the urethra is 5 times longer in males than in females. In males, it's divided into 3 segments. In females, it's one short tube. The urethra is a common duct for urinary and reproductive systems in males, whereas in females, 2 systems are entirely separate. So one of the last things that we talk about is the body's ability to regulate and handle waste management. So we use a series of systems here. We'll use one, the GI tract, so the digestive system, to help remediate and remove waste. Remember sweat when we did the integument system. We talked about sweat being a form of dilute urine, another way of removing waste products, urea and things like that. We talk about the lungs, removing waste, helping the cardiovascular system with the blood, of adding nutrients and unloading waste. So again, another aspect of waste management. The liver plays a major role in waste management, as does the buffers of the body. So buffering the body with acid base is going to also help mitigate waste management. Which brings us to our second to last slide. Aside from our focus on homeostasis, we deal with aging in the urinary system. And this is pretty interesting because a lot of people don't realize how important their kidneys are, and especially what happens to them as we age. So as we age, our kidneys shrink in size. They lose about 1/3 of their mass. If you think about that, that's a big loss. 33% of our mass of our kidneys disappears or reduces in function. We experience a decrease in blood flow. Those glomerular vessels tend to be damaged over time, or they decrease in number, especially damaged with people with hypertension or high blood pressure. And we experience a decrease in filtration, which is why as we get older and disease ensues, we see a lot of elderly undergoing a procedure called dialysis. So what dialysis is basically a treatment that does some of the things that your healthy kidney should be doing. It's basically needed when your own kidney can no longer take care of your body's needs. Now, we typically set a threshold for this. So, dialysis is typically needed when you're towards an end stage kidney failure. Usually, by this time, you lose about 85 to 90% of your kidney function and have a glomerular filtration rate of less than 15. So, think about this. When your kidneys fail, dialysis keeps your body in balance. It does this by removing waste, salt, and any extra water to prevent them from building up in the body or causing edema. It also keeps a safe level of certain chemicals in your blood, things like potassium, sodium, and bicarbonate. It also plays a major role in helping to regulate blood pressure. So dialysis can be a useful and helpful tool when regulating and monitoring the elderly when they suffer from end-stage kidney disease or kidney failure. Our last slide that we talk about is going to have our focus on homeostasis, how the urinary system contributes to all body systems. So we look at our center board. Kidneys regulate volume, composition, and pH of body fluids by removing wastes and excess substances from blood and excreting them into the urine. The ureters transport urine from kidneys to the urinary bladder, which stores urine until it is eliminated through the urethra. So we can see how the digestive system kicks in and helps. Kidneys help synthesize calcitriol. This is going to help absorb our dietary calcium. Our reproductive system, in males, the urethra extends through the prostate, and the penis is a passageway for semen as well as it is for urine. Endocrine system, all those hormones we talked about. Erythropoietin from the kidneys for red blood cell production. We talk about calcitriol, the active form of vitamin D. In the nervous system, we talk about gluconeogenesis, which provides glucose for ATP production in neurons, especially during fasting. Muscular system, kidneys help adjust levels of blood calcium needed for contraction of muscles. Skeletal system, same thing. We're monitoring and adjusting levels of blood calcium and phosphate needed for building that bone matrix. In the integumentary system, kidneys and skin both contribute to synthesis of calcitriol, which is the active form of vitamin D. We can move over to the cardiovascular system, increasing or decreasing the reabsorption of water filter from blood. The kidneys are going to help adjust blood volume and blood pressure. We talked about the renin-angiotensin system, and finally some bilirubin. Bilirubin from hemoglobin breakdown is converted to a yellow pigment called urobilin, which is excreted in urine. And when you do urinalysis, you will check for urobilinogen and which level should be normal in normal urine. Our lymphatic system, by increasing or decreasing the reabsorption of water filtered from blood, the kidneys help adjust volume of interstitial fluid and lymph. Urine flushes microbes out of the urethra. So that takes care of our urinary system. I hope you found this lecture to be helpful.